is entitled That. Thanks.
Right, I suppose we all know that temperature is a very important environmental variable because it's fluctuated over evolutionary time, but it's also increasing with climate change. In this graph, you can see the blue line represents observed sea surface temperatures on the Great Barrier Reef from 1871 onwards. And over that time period, we have already seen an increase of about 0.4 degrees centigrade. The green and red lines indicate projected temperatures in the year 2100 based on models assuming low and high greenhouse gas emissions. And you can see that the predictions are that further temperature increases will occur. Now, this is a problem for corals because corals will bleach when they experience temperatures higher, 1 or a few degrees higher than they used to. Now, the coral animal associates with a wide range of microbial organisms. The single-celled algae, the zooxanthellae, which live inside their cells, but also with bacteria and archaea and viruses. When corals bleach, they lose the zooxanthellae, and if bleaching conditions persist, they will eventually die. So that means that corals will have to adapt, or they will likely suffer considerable mortality. Now, in my talk today, I'd like to address 4 topics. First, I'd like to provide some evidence of past adaptation of corals to temperature. After that, I'd like to present some results from my lab on the relative adaptive capacity of the coral animal versus its associated zooxanthellae. Oh, didn't even touch the button. Next, I'd like to talk a little bit about the potential relevance of new mutations as a source for selection. And finally, I'd like to present some preliminary results from my group on the potential usefulness of some management options. I don't know why this is going. Can we stop this? It's automatically going forwards. I can't talk that fast. So, sorry? It's only on the screen, isn't it? They fixing it? Okay, just go up one. And so finally, we're trying to investigate the usefulness of a management strategy to aid in adaptation of corals to temperature. But before I start, I'd like to acknowledge my collaborators and students who have contributed to the data that I will present today. Now, what is the evidence for past adaptation to temperature in corals? Several studies have shown that geographic differences exist in the upper temperature tolerance limits of corals, and that these correlate with ambient temperatures. If such differences are maintained after we acclimate the corals to the same environmental parameters, usually in the laboratory, this is usually taken as evidence that corals have adapted to local environmental conditions. For example, on the right-hand side, I've summarized an experiment that was done at Ames a few years ago, where we compared upper thermal tolerance limits between 3 coral populations along a temperature gradient on the Great Barrier Reef. And the shading in the circles indicates the health status of these corals at the end of the experiment. And you can see that the corals from the hottest area were the most tolerant. They were still healthy at the end of the experiment. The ones from the intermediate temperature were partially bleached, and the ones from the coolest region were severely bleached or dead. So these results show that selection has led to differences in the genes and the gene frequencies of these geographic populations. And in the next few slides, I will show you that such genetic differences can occur both in the zooxanthellae and the coral animal. First, the zooxanthellae. This experiment was done by one of our PhD students, Emily Howells. And for this experiment, we collected 2 populations of the same species of zooxanthellae, Symbiodinium C1. And we collected those populations from 2 distinct thermal environments, Magnetic Island and the Whitsundays. Following the coral mass spawning event, we reared Acropora millepora larvae, and these larvae had the same genetic background. They were raised from the same cross, involving a small number of parental colonies collected from a single population. We settled the larvae and infected the juveniles either with the Magnetic Island or the Whitsunday zooxanthellae, and then subjected them to heat stress in the laboratory. And what we found was that the juveniles, which associated with the zooxanthellae from the hotter area, were the most temperature tolerant, and the ones from the cooler area were less tolerant to high temperatures. And this provides evidence that there are some genetic differences between these zooxanthellae populations that result in the difference in the temperature stress response in the coral holobiont. In a similar experiment, we didn't vary the source of the zooxanthellae, but we varied the source of the coral animal. And we reared juveniles from the Keppel Islands in the southern GBR and Magnetic Island in the central GBR. We infected those juvenile corals with a single population of zooxanthellae, clay D zooxanthellae from Magnetic Island. We grew those corals out for a couple of months, brought them back into the lab, acclimated them, and subjected them to heat stress. And this graph, the solid lines in the graph, shows the response of those two groups of juvenile corals. And you can clearly see that there is a significant difference in the response between those groups of corals. And that can be explained by genetic differences occurring between these geographic populations of corals. Now, while it is important to understand how and to what extent corals have adapted in the past, the $64 million question really is, can further adaptation to temperature occur and how fast? In other words, if the bell curve on the left-hand side represents thermal tolerance within a coral population today, can selection push that population to have an increased thermal tolerance to the right-hand side in this graph? And the way to estimate that, to examine that, is to estimate the portion of variation in thermal tolerance that has a genetic basis, and we call that the heritability. And you need to know that trait variations of phenotypic variants has a genetic and an environmental component. For example, if we look at human height, some of the most important environmental components underlying the phenotypic variance in human height are diet and quality of healthcare. For example, if you eat very poorly as a child, you will never reach, or you are unlikely to reach full potential in terms of height as an adult. The genetic components are the variants, the alleles of hundreds of genes. So if you are a child in a family of only short people, you're unlikely to grow very tall, even if you eat very, very well. So selection can only act on this genetic component that underlies phenotypic variation, and adaptation can only occur through selection. Now, in a series of experiments, we estimate the heritability, this genetic component underlying variance, of a range of thermal tolerant traits in Acropora and millepora. And the way we did that is we estimate, or we quantified the variance in thermal tolerance between clonal branches from individual coral colonies and compared that to the variance in thermal tolerance between genetically distinct coral colonies. And by a simple subtraction, because the variation between genetically identical branches can only be due to environmental factors. So by a simple subtraction of that portion from the total variance, in other words, the variance among colonies, will give us an estimate of the genetic variance underlying thermal tolerance. Now, I don't have time to go through these experiments in detail, so I will summarize it with this slide. But the preliminary work showed that the general trend was that traits which we assumed were mainly under the control of the zooxanthellae, or more or less under equal control of the zooxanthellae and the coral host, had relatively high heritabilities, while traits that were mostly under the control of the coral animal, and we used expression level of genes encoded in the coral genome, had generally lower heritabilities. This, in combination with the short clonal turnover times of the zooxanthellae, which on average are about 2 weeks, it theoretically allows for rapid adaptation by selection on existing levels of genetic variation. By contrast, rates of evolutionary chain in host traits are likely to lag behind those of the zooxanthellae because of those lower heritabilities, but also because of the longer generation times that corals generally have. In other words, the conclusions from these experiments was that the zooxanthellae are likely to be able to adapt faster than the coral animal. And this is a very important finding, but keep in mind that this is based on a relatively small number of traits, and we really need to validate whether that holds when we look at other traits as well. Now, with this slide, I'd like to show you how we can use estimates of heritability to model the response to selection. And I used the heritability of photosynthetic efficiency here, which we found was 0.5. And the response to selection is given by the breeder's equation. It's the heritability multiplied by the selection differential. I'll explain that to you. So what I've plotted in this graph is the reduction in the maximum quantum yield, the photosynthetic efficiency, of the 20 colonies we used in the experiment, and at 32 degrees relative to the control temperature. And the mean reduction in FV/FM was 27.4%. Now let's assume that the 6 most tolerant individuals within that population contribute to the next generation. This is very hypothetical. Now we can have a look at the reduction in FEFM in those 6 individuals, which was 23.1%. The selection differential then is the difference between those two figures is 4.3%. And we can simply fill that in the breeder's equation to get the theoretical response to selection, which is a reduction in the drop of FEFM by 2.2%. In other words, the selected generation would have a reduction of 25.2% rather than 27.4%. Now, this doesn't sound like much, perhaps, but if you think about the fact that clonal generation times in zooxanthellae can be as short as a few days, it's actually quite a significant change. So this sort of heritability work I just discussed basically only takes into account the existing levels of genetic variation within the population, and it doesn't take into account any new mutations that may arise and selection on those new mutations. And in general, new mutations are usually considered unimportant in terms of the rates of adaptation of the timescales that we're looking at in terms of climate change and ecological change. But this is actually largely untested, and I'd like to challenge this. So mutations can occur during meiosis. Meiosis is the cell division that leads to the formation of gametes, basically sexual reproduction. And if a mutation takes place during meiosis, that mutation is passed on only to a small number of gametes. And it's maybe a bit hard to see, but I've indicated that by the green and red colors. Those little circles are the gametes. However, mutations can also occur during mitosis. Those are somatic mutations. And imagine that somatic mutations occurs here in a coral polyp. As that coral keeps on growing and budding off new polyps, that mutation will be passed on to the rest of the coral colony. And when this coral spawns, all the gametes will have that mutation as well. Furthermore, corals don't have a separate germ and somatic cell line, so even in this polyp where the mutation occurred, theoretically that mutation can be passed on to the offspring. In terms of somatic mutations in zooxanthellae, I think they may be very important as well. Because when the zooxanthellae are inside the host, they only reproduce asexually. And so hence those mutations, if they have a large advantage, selective advantage, they can be selected upon and they may replace the existing population of zooxanthellae. And interestingly, mutation rates of beneficial spontaneous mutations have been estimated for a range of single-celled algae. And they are on the order of 10 to the minus 5 to 10 to the minus 7. Now, assume that these rates are similar in zooxanthellae. What does that mean? We know that zooxanthellae occur at quite high densities in corals, about 1 million cells per square centimeter. So, in theory, 0.1 to 10 mutations could occur per square centimeter of coral per clonal generation. Now, I think that probably the surface area of a coral varies between maybe several tens to several hundred of square centimeters, maybe even more than 1,000. So these are theoretically quite significant numbers. And again, don't forget, clonal generation time to zooxanthellae can be fairly short. And this is an area that hasn't received any attention as far as I am aware, but I think we really should look into the role of new mutations as a source for selection and potentially adaptation. Now finally, I'd like to briefly present some preliminary experiments we have done to examine the usefulness of a management strategy to help corals adapt. And several of such strategies have recently been proposed, and most or all of those are largely untested. So some of those include reseeding of damaged reefs with cultured tolerant coral genotypes, selective breeding of more tolerant genotypes, and the introduction of beneficial alleles into certain populations by translocations of coral colonies harboring such beneficial alleles. And of course, before we can even begin to think of implementing such strategies, we need to know a lot about, first of all, the genetic basis of stress tolerance, and also the impacts of translocation on the receiving population. And this is the latter is the area where we've done a little bit of work in, and I will present it to you now. So we looked at translocations from the central GBR of Island to the Keppel Islands. And the first question we asked, can colonies from those two regions interbreed? Because the reasoning behind this is that those translocated colonies will interbreed with the local population and as such introduce beneficial alleles into that population. And yes, we found that they bred with very high fertilization success. In fact, we didn't find any significant difference in fertilization success between the purebred within population and the regional hybrid fertilization. However, we found that the rate of development of the embryos was significantly faster in the purebred crosses. Sorry, I need to get a sip of water. Next, we looked at a few proxies of fitness, because the desired outcome of these strategies is that the hybrids are fitter than the native purebreds. You want to improve the performance of that population. And as a proxy for fitness, we looked at both survival and growth. And here I've plotted survival of the native purebreds in green, the hybrids in blue, and the purebreds from Orpheus Island in red. And you can see that the hybrids are significantly show lower survival compared to the purebreds, so that's not good. We also looked at growth, and quite interesting, there we found that the hybrids actually grow faster than the Keppel Island purebreds, so that's some evidence of hybrid vigor there. Now this year we will repeat these experiments, and what we really want to quantify is thermal tolerance, because what we would hope is that the hybrids between Orpheus Island and the Keppels show improved thermal tolerance compared to the Keppel Island purebreds. And luckily this brings me to my conclusion slide, because I've got a cough. My conclusions are a little bit boring, but it's because very surprisingly little research has been done in the area of potential for adaptation of corals to climate change. So we know that mechanisms for adaptation exist, but it's still uncertain whether these may be sufficient for corals to cope with the rapid rate of warming. So I think we should really direct more research effort towards this kind of research. Thank you very much.